Hello students, welcome to Trunkie Academy. Today we'll study another topic. Like yesterday we discussed about so we'll start the new chapter. Okay, so that is the cell, and the cell is as you have already studied in the previous classes, cell is the structural and functional unit of life. Okay, and to start with this cell, first we'll study about the prokaryotic cells, and you know that when you studied in previous class in the lower classes in the class 9 also you studied about the cells, that there are two kinds of cells, prokaryotic and eukaryotic cells. Prokaryotic are the primitive type of cells where the nucleus is not prominent and the chromosomes are dispersed in the cytoplasm. So okay, but this is also this topic is very important for the exam and we have to study in detail. Okay, so prokaryotic cells are found in bacteria, blue-green algae and the mycoplasmas, and PPLO, that is the pleuropneumonia-like organisms. Okay, so these are the type of organisms where we find these prokaryotic cells, primitive type of organisms. Okay, so this is the what do we call this? Prokaryotic cells. Okay, so blue-green algae, bacteria. Okay, blue-green algae are also called cyanobacteria. Okay, mycoplasma and PPLO, pleuropneumonia-like organisms. So these are the organisms. Okay, and they are unicellular organisms and they have these cells as prokaryotic cells. Okay, now when we say blue-green algae, these blue-green algae are capable of carrying out photosynthesis. Some are, and some are they can synthesize their food with the help of a process called chemosynthesis. Okay, so they are pleural pneumonia. This is the picture. Okay, PPLO, and the prokaryotic cells are generally smaller and multiply more rapidly than eukaryotic cells. Okay, so when you compare the size of the cell with eukaryotic cells, we find that the prokaryotic cells are very smaller as compared to the eukaryotic cells. Since they are smaller, they can divide rapidly than the eukaryotic cells. Okay, so they may vary greatly in shape and size. We say so many different variety of bacteria are found, and bacteria have invaded all different types of habitat, and they can found on all different types of habitat like hot springs or in the glaciers or in the deep into the soil or in the water. Okay, and in the desert. Okay, in dry conditions. So different types of habitat they have invaded, and they are adapted for that mode of their habitats. Okay, so how many basic shapes are there in bacteria? There is the 4, depending on there is shape. They are classified into 4 types. Bacillus rod shaped, and the coccus the spherical, and the vibrio comma shaped, and the spirillum is a spiral. Okay, the spiral, the spring shape. Okay, so these are the 4 basic shapes of the bacteria based on their 4 categories types based on the shape they are classified. Okay, shape there are 4 types. Okay. When we say bacillus, the rod shaped, okay, like lactobacillus and all. Coccus, like streptococcus, pneumococcus, they are the coccus is spherical, and they may be like joined. Okay, they may be free or they may be joined to the diplococcus. Okay, and vibrio, like comma shape. Vibrio means comma shape. This is like this one. They are like this. Okay, comma shape, or you can say rather you can like this. So this is like this, and so are called vibrio. Now spirillum, they are spiral or little spring shaped. Okay, and these are the 4 types. Organisms range. Now see, typical eukaryotic cells, they range from 10 to 20 micrometer. Okay, in animal cell. So 10 to, and now you see the typical bacterial cells very small. Okay, almost 10 times smaller. Okay, so 1 to 2 micron. Okay, the range, and PPLO is even smaller, 0.1 micrometer, and viruses are 0.02 to 0.2 micrometer. So this virus is the smallest. Okay, so generally, but remember, this virus is not the prokaryotic cells. Okay, virus has no actually cell. Virus is a special kind of structure that we'll study separately. Eukaryotic cells, we are here talking about the sizes, not about the prokaryotic or eukaryotic. The size of the eukaryotic cells ranges from 10 to 20 micrometer, Typical bacterial cell ranges from 1 to 2 micrometer, and PPLO it is 0.1 micrometer. Viruses 0.02 to 0.2 micrometer. Generally, prokaryotes have a cell wall surrounding the cell membrane. Okay, so that the cell wall is present. The fluid matrix filling the cell is the cytoplasm. So that you know very well that when you studied in class 8 also, what is cytoplasm, what is nucleus, and what is the plasma membrane and cell membrane, that we have already studied in previous classes. So fluid matrix is present in the cell, and that is the cytoplasm. And there are no cell wall well-defined nucleus. So that is the main feature. Why it is called a prokaryotic. Pro means primitive and karyon means nucleus. So primitive nucleus, primitive type means where there is no well-defined nucleus, or there is no prominent nucleus, or the nucleus without cellular membrane, nucleus membrane, and then there is no prominent nucleus. The nucleus membrane is absent, and the prominent nucleus is absent. No well-defined nucleus is present, and the chromosomes are dispersed in the cytoplasm. So it is called the prokaryotic. Okay. Now the genetic material is basically naked. So like we have seen in case of the eukaryotic cells, the genetic material with the nucleosome, then it is the chromatin fibers there and well packed in the chromosome, isn't? Nicely with the help of the there is an octacore. Okay, structure is there on which the DNA are wounded. Okay, that is called nucleosome. But here the histone protein, octamer histone protein is found in case of the eukaryotic cell. Histone protein. Okay, that is not found in case of the prokaryotic cells, and it is the naked. Okay, genetic material naked. In the prokaryotic, the genomic DNA stands for single chromosome, that is circular DNA. So when we observe in case of the DNA, that is it is a linear. It may be linear in case of eukaryotic cell, but in case of the prokaryotic cell, it may be like this, and it is the circular. So it is continuous. Okay, the circular DNA is found, and it is called plasmid. Also, okay. Now, in addition to genomic DNA, the many bacteria have small circular DNAs outside the genomic DNA called plasmids. So try to understand this. The genomic DNA means the genome. Okay, the outside the plasmid, outside the genomic DNA means the chromosomes like the structure, the main DNA, which is actually the responsible for the expressing the characters. Okay, in the progenies. And apart from the genomic DNA, there is another kind of circular DNA that is called plasmid. Okay, so in addition to genomic DNA is another DNA that is present in the cytoplasm, that is called the plasmid. Circular DNA is found like this, plasmids. Okay, the bacterial DNA, then the plasmid circular DNA. So this is the present in this. This is a type of DNA present in the bacterial cell. Okay. What is meant by naked genetic material? Means the genetic material not enveloped by nuclear membrane. Okay, so that means there is no nuclear membrane, there is no prominent nucleus, there is no distinct nucleus present in the cytoplasm, and the genetic material is naked, that is dispersed in the cytoplasm. Okay, there is no nuclear membrane. Now, what are plasmids? These are the additional small circular DNAs. Like we have seen, the DNA is present in the chromosome, and that chromosome is present in the nucleus. But apart from the nucleus, if the chromosomes or DNA is found in cytoplasm, okay, apart from the nucleus, like in case of the chloroplast or mitochondria, where they have their own DNA, so that we will call the extra nuclear DNA, okay, or cytoplasmic DNA, or extra. Okay, so that is the likewise here it is the apart from the genomic DNA, they are the DNA which are called the plasmids, and they are circular in shape, and they are the circular DNA and called plasmids. Okay. Now the plasmid DNA confers certain unique phenotypic characters to such bacteria. Plasmids containing bacteria are resistant to antibiotics. Okay, so that means they are unique phenotypic characters. Okay, means when the progenies or new offsprings are produced, okay, so they will have some unique features, unique characters. Okay, and plasmids containing bacteria are resistant to antibiotics. Plasmids, because they have got some kind of gene that confers and that provides resistance to the antibiotics. Okay, antibiotics are the medicines, as you know, that I discussed already. No, you will study in class 12. Okay, in detail, but you have already studied in class 9 also a little bit of this antibiotic. Antibiotics are the medicine or chemical substances that act against the life of pathogens. Biotics, bio means life, and anti means against the life of the pathogens, to kill those bacteria. Okay, and in case of the plasmid containing bacteria, are they resistant to antibiotics. Nuclear membrane is found in eukaryotes, so they are called eukaryote. U means true, and karyon means the karyotes. The word is derived from the karyon, and the karyon is the nucleus. So true nucleus is present in case of the eukaryotes, because nuclear membrane is present and the true nucleus is found, distinct nucleus is found, prominent nucleus is found, and there is the chromosomes inside the nucleus. So ribosomes are present in both prokaryotes and eukaryotes, but ribosomes are common to both. Ribosomes are another cell organelle which is very, very important in involved in the synthesis of protein, polypeptide chain. Okay, so it is found in both prokaryotic and eukaryote. Mesosomes are the characteristics of prokaryotes. Mesosomes are found in prokaryotes but not in eukaryotes. Okay, so what happens when which cell organelle are present in both prokaryotes and that is the ribosomes, but mesosome is only present in eukaryotes. In case of the eukaryote cells, we find mitochondria. Okay, but here in case of prokaryote, it's the mesosomes. Okay, ribosomes is the common to both. Now, what are mesosomes? Membrane extensions into the cytoplasm called mesosomes. Okay, so that membrane, the cytoplasm, which is the membrane, the cell membrane which is there in the bacterial cell, that has got some extension into the cytoplasm. For example, if I will draw here, if this is the cell membrane and this has got some extensions, okay, and again here, then this is the extension of this, then here again this membrane get the extension like this. So this extension is the mesosomes. Okay, so the membrane extends into the cytoplasm. So that is the mesosomes. So roughly, I have explained with the help of this diagram. Okay. Now, cell envelope and its modifications in prokaryotes. Now, bacterial cells have chemically complex cell envelope. Bacterial, okay. As we have observed in other plants or eukaryotic plants or green plants or higher plants, or in higher animals, okay, we find that the enveloping and the covering is little complex, and chemically it is a little different also. Okay, so the cell envelope consists of tightly bound three-layered structure. That the outermost layer is the glycocalyx, okay, made of the glycocalyx, and followed by the cell wall and then the plasma membrane. So finally, we see that means the last and the innermost is the plasma membrane, which is also called cell membrane, which is found in all living cells, okay, or in all cells of the living organisms. Then in the cell wall is there, okay, and then is the glycocalyx. The outermost layer is the glycocalyx, and which has some characteristic. It is characteristic to the feature of the bacteria or prokaryotic cells. Okay, the cell wall is present, and then there is the glycocalyx. Okay, even the composition and the chemical substance that is present in the cell wall, in the making of the cell wall, or for manufacturing, or the constituent part, or the constituent chemical of the cell wall is also different from the cell wall of the green plants. Okay, so first is the plasma membrane, okay, and then is the cell wall, and then is the glycocalyx. Now what we'll see, cell envelope consists of three regions: the glycocalyx, the cell wall, and the plasma membrane. Okay. Although each layer of the envelope performs a distinct function, they act together as a single protective unit. All these three layers have got their three different functions. They have their own, they have their functions, okay, and they have their own function, but together they are acting as a unit for protection of the bacterial cell. Okay. Now bacteria can be classified into two groups based on their chemical composition of envelope and response to staining. Okay. So what happens when we say response to staining means when we are staining, which type of stain they are retaining, or which type of stain they are taking, okay, and which type of stain they are not taking, not getting stained with. Okay, so depending on that, there are two groups. Okay, and chemical envelope or chemical composition of envelope also, based on that there are two groups. This staining procedure developed by Graham. Okay, Graham is the name of the scientist who developed this technique. Okay, so when he was staining the bacterial cell, he found that some bacteria are taking one particular type of stain or dye, and another group of bacteria are taking another type of dye or stain. So depending on that, so he divided into two types, okay, gram positive and gram negative. Bacteria based on the response of the staining, gram positive and gram negative, okay. So this is the, now we see the bacteria that takes up the gram stain is called the gram positive, and the bacteria that do not take up the gram stain is called gram negative, okay. So there are two categories of bacteria based on the response to staining, okay. So staining, while staining, we find that some group of bacteria they take up the stain and some do not take. That is a gram negative, okay. So glycocalyx differs in composition and thickness among the different bacteria. In different variety of bacteria, the glycocalyx, which is the outermost layer of the bacteria, this differs in composition. Okay, the composition of the glycocalyx may be different. Also, the thickness of the glycocalyx differs in different group of bacteria. Okay, so now gram positive envelope. Okay, lipoticoic acid is there. Ticoic acid, lipoticoic acid and ticoic acid, peptidoglycan. Okay, peptidoglycan is one of the kind of the chemical substance. Okay, peptidoglycan, and there is a periplasmic space is there in the plasma membrane. So innermost layer, right after the cytoplasm or protoplasm, is the plasma membrane, which is common to all living cells of the all living organisms. And after that, the periplasmic space is there. You can see, and then peptidoglycan. So this is the cell wall. This part is the cell wall, peptidoglycan. So cell wall is made up of the peptidoglycan. Okay, and after that you can see there is a ticoic acid and the lipoticoic acid. So with the one with the having this ticoic acid and lipoticoic acid, they are the gram positive. Okay. Now in case of this, you can see the side chain. Okay. The porine. These are the plasma membrane, then the periplasmic space, okay, then and peptidoglycan, the outermost membrane, okay, then the lipopolysaccharides and the porins, the channels through which substance enter, okay, and this is the gram negative envelope. So they have got two layers, you can say the more complex layer as compared to the gram positive, okay. So they have got the another layer of the side. They have got some layer of these lipopolysaccharide, okay, and this is so in the central in the middle you can find the peptidoglycan, periplasmic stain and peptidoglycan. So this is a gram negative, and it will not take the gram stain. Okay, glycocalyx, it could be a low sheath called the slime layer, okay, it can it may be thick and tough like called the capsule. In some cases it is like slimy mucus like in the layer, okay, and low sheath, okay, semi-solid or little flowing, okay, or it can be thick, tough, little dry and like a capsule, okay. So it depends on the type of bacteria. The cell wall determines the shape of the cell, okay. So cell wall actually determines the shape of the cell of the bacteria, okay. So cell wall provides a strong structural support, okay, because cell wall is hard, rigid, and that provides the shape of the bacterial cells to prevent the bacterium from bursting or collapsing, because they are growing in different types of medium that may cause the swelling of the bacterial cells or some other kind of changes in the shape. To prevent that, the bacterial cell wall provides the structural support and thus maintain the shape of the bacterial cell. Okay. Now, what determines the shape of the cell is the cell wall of the islet. This now is the plasma membrane is semi-permeable in nature and interacts with the outside wall. And this is an important point that we have already studied in previous classes. Okay, in class 9 also you might have studied this. Plasma membrane is semi-permeable or selectively permeable membrane, which we have already studied in class 9. And this is a very important point, because this plasma membrane or cell membrane, which is semi-permeable or selectively permeable, allows the entry and exit of the substance whichever is required for the cell. Okay, and this membrane is structurally similar to that of the eukaryote. So I told you the cell membrane or plasma membrane is common to all cells of the living organisms, which is found in all living organisms. So a special membranous extension of the plasma membrane into the cell cytoplasm is mesosome. I told you at that time, mesosomes are the extension of the plasma membrane into the cytoplasm. Okay, extensions are in the form of vesicles, tubules, or lamellae. So if the extension going inside the cytoplasm, it can form a spherical vesicles or like a tubules or like lamellae. Okay, like a small flat. Okay. Now the cell wall formation, DNA replication, and its distribution to total cells, and helps in respiration, secretion process to increase the surface of the plasma membrane for absorption of nutrient and enzymatic content. These are the functions of the mesosomes. So very important here you can say. So it helps in respiration, as I told you. When I told you the mesosomes are found in prokaryotic cells, mitochondria is found in eukaryotic cells. I told you that time, because the site of respiration in case of the eukaryotic cells, the mitochondria, whereas in case of this here, we can see the mesosomes are involved in respiration. But not only in respiration, this function as also it plays an important role in the cell wall formation. Okay, DNA replication, and it also plays an important role in DNA replication and its distribution to daughter cells, and secretion process or different types of secretory process, and to increase the surface area of the plasma membrane for absorption of nutrients, and there are some enzymes contained in the mesosomes. So these are the functions of the mesosomes. Okay, that is only characteristic of this bacterial cells, not found prokaryotic cells, not found in eukaryotic cells, most of the mesosomes. Okay, in some prokaryotics like cyanobacteria or blue-green algae, there are other membranous extension into the cytoplasm called chromatophores, which contain pigment chromatophores. Okay, it is also the membranous structure into the cytoplasm from the cytoplasmic membrane, and that is the not mesosome, that is the chromatophores, and which contain pigments. Chromato means the main color, and the phores means the substance. Okay, the structure. Now chromatophores are the structure that carry the pigment. Okay, so that is they are present in the cyanobacteria. Okay, the prokaryotic cells of the blue-green algae, so not in bacteria. Okay, but in the cyanobacteria, you can see chromatophores. Okay, bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension from the cell wall called flagella. Okay, some bacteria are motile, they can move. Okay, and some are non-motile. Okay, if bacteria are motile, they have thin filamentous extension from the cell wall. Okay, filamentous thread-like filamentous structure called flagella, and with the movement of this flagella, they are able to move. Bacteria show a range of in the number of arrangement of flagella. Some bacteria, they have got 2 flagella, some has 1, some is all over the body. So depending on the type of group or type, bacteria show a range in the number and the arrangement of flagella over their body. Okay, so this is the flagella, the filamentous. Okay, cell-like structure extending from the cell wall. Okay, so that is the flagella, which helps in the motility of the bacterial cell. Bacterial flagellum is composed of a filament, a hook, a basal body. Now, if we observe very minutely and we observe very closely, okay, the structure of this bacterial flagellum, so we find that there is a long thread which is outside long, that is the filament. Okay, and at the basal region there is a hook. Okay, and right below the hook there is a basal body. Okay, and which is anchored into the membrane. Okay, under the plasma membrane. Okay, and then the plasma membrane gives rise to the hook, and then there is the filament. So this is the structure of the the structure, how this bacterial flagellum is formed of and what it's composed of. Okay, so filament, hook, and basal body. Basal body is embedded in the plasma membrane of the bacterial cell. Okay, now the filament is the longest portion and extends from the cell surface to the outside. So filament, obviously, because this is from the basal, from the region, it extends from the cell to the outside. Okay, it's a long filament-like structure. Okay, filament is, and that filament is the longest portion. Besides flagella, pili, fimbriae are also surface structures of the bacteria but do not play a role in motility. Apart from the flagella, there are some certain other structures, filamentous structures are found in the bacteria. Other of pili, besides flagella, pili and fimbriae. So that is called pili or fimbriae are found. There are also a kind of filamentous structure on the surface of the bacteria. But these pili and fimbriae do not provide motility, do not play a role in movement of the bacteria. They have got some other important role, like pili, it plays important role in reproduction of the bacteria. Okay, so the pili are elongated tubular structures made of a special protein. Okay, pili are the tubular structures, a filamentous structure are made of a special protein in the bacteria. Okay, so this is the pili. You can see the red one is the pili. Okay. So what is the longest portion in the bacteria? Is in the flagellum. This is the filament. Okay. Now the fimbriae are the smallest bristle-like fibers sprouting out of this cell. Fimbriae are the bristle, very finer thin bristle-like the bristle of the toothbrush. Okay. They are coming in a group. Okay, not one or two bristles. So that's why you call it the bristle-like fibers. Okay. So here you can see the bristle-like. This is the fimbriae. Okay, fimbriae. Can you see that? This is our fimbriae. Now, these fimbriae, in some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues, because since they can be blown away or be, just by moved away by the water current. Okay, so they need to attach to some substratum like rocks or in streams in the river or some kind of small streams, or to the host tissues. So these fimbriae helps them to attach to the host tissues or to some substratum such as the what we call this rocks, etc. Okay, the rocks in the stream here you can see they are attached with the help of this fimbriae. Okay, some bacteria. Host tissues means we are talking about the tissues of the other living organisms where they are attached. Okay, now ribosomes. As I told you, ribosomes is the structure or cell organelle common to both prokaryotic and eukaryotic cells. Okay, ribosomes are found in both prokaryotic and eukaryotic cells. Okay, in prokaryotic, ribosomes are associated with the plasma membrane of the cell. They are associated with plasma membrane of the cell in case of the prokaryotic. But in case of the eukaryotic cells, ribosomes are in the cytoplasm, not associated with the plasma membrane, and some are present on the surface of the endoplasmic reticulum, and hence this endoplasmic reticulum is called the rough endoplasmic reticulum that we have already studied in previous classes. So there are two kinds of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, so ribosomes are present in both eukaryotic and prokaryotic. In case of the prokaryotics, these ribosomes are associated with the plasma membrane. They are about 15 nanometer by 20 nanometer in size. Ribosomes made of two subunits, 50S and 30S units, which when present together from 70S prokaryotic ribosomes. Okay, ribosomes made of two subunits, 50S and 30S units, which when present together from the not from its form FORM, not from together from 70S. Okay, S is a sweat bug unit. Okay, it's a kind of the which actually it is denoting to denote the size actually. When we centrifuge, that depending on that we'll study later on in the other topics. So ribosomes made of two subunits. Okay, 50S. In case of the eukaryotic cells also there are two subunits. Okay, but it is bigger in size. Okay. Now ribosomes are the site of protein synthesis in both in prokaryotic and eukaryotic cells. Okay, so what is the size of the ribosomes in prokaryote? It is 15 to nanometer by 20 nanometer. Okay, so this is the size of the ribosomes in prokaryotic cells. Several ribosomes may attach to a single mRNA to form a chain called polyribosomes or polysomes. Sometimes what happens, the mRNA, messenger RNA, which has carrying the message given by the DNA, and that is now going to be translated into amino acid and polypeptide chain. So ribosomes are attached with the mRNA and in a long chain fashion. Okay, that is called the polyribosomes. Okay, one by one they are attaching with the ribosomes and form a long chain. Okay, it's called the polyribosomes or polysomes. So that is also found in bacterial cells, which is not found in case of the eukaryotic cell. The ribosomes of a polysome translate the mRNA into proteins. Okay, so that kind of structure is found. Polysomes or polyribosomes is the common feature in the prokaryotic cells. Okay, so ribosomes, which is ribosomes of the polysome or polyribosome, translate the mRNA into protein or amino acid and then protein. Now, inclusion bodies. This is another structure that is found in the prokaryotic cells. Reserved food materials in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Some of the structures are found in the prokaryotic cell that store the food and that are present in the cytoplasm in prokaryotic cell. Okay, so these inclusion bodies are not bounded by any membrane system. Okay, and lie free in the cytoplasm. They flow freely in the cytoplasm, and they are the granules-like, but there is no any kind of membrane-bound structure. Okay, so they are just the granules or some structure. Okay, and they are lying freely in the cytoplasm. Phosphate granules, cyanophycine granules, and glycogen granules are the examples of inclusion bodies. These are the storage. They form actually storage granules and stored food material that lie in the cytoplasm. Okay. Now, gas vacuoles are found in blue, green, purple, and green photosynthetic bacteria. Gas vacuoles. Okay, large vacuoles. Okay, one which is the vacuoles that is contained in the cytoplasm, and there's some are bigger in size in case of these bacterial cells. They contain gas. They contain gas. In case of the blue, green, or purple or green photosynthetic bacteria, so gas vacuoles are found. Okay. The structure of prokaryotic cells that separates the interior of the cell to the exterior of the cell. Okay, so that is the cell wall or plasma nucleoid or cell membrane, or it is a slime layer. It is a cell membrane. Okay, so it separates the interior of the cells to the exterior of the cell. That is the cell membrane. Cell membrane is the limit to the cytoplasm. Okay, so cell membrane is the structure which separates. Now, ribosomes of a polysome translate mRNA into protein or amino acids. Okay, so it is a protein. Okay, not sugar or that. Now, genomic DNA of bacteria is naked, linear, and double-stranded, membrane-bound, circular, or double-stranded, membrane-bound, circular, and double-stranded, or naked, circular, double-stranded, or naked, linear, single-stranded. Very simple. Okay, but very important question. Okay, it is the naked and circular, circular and double-stranded. DNA is a double-stranded structure and mRNA is a single-stranded. Now, circular DNA outside the genomic DNA in bacteria is called plasmid. Viroin, this is the plasmid. Okay, so that is the after. Apart from the genomic DNA in the cytoplasm, we find some circular DNA that is called plasmid. Okay. Now, reserved food materials in prokaryotic cells are stored in. It is the inclusion bodies. Okay, inclusion bodies. Okay, and that there is no membrane. They are just lying in the cytoplasm, and they have some stored food material. Thick and tough. Thick and tough. Okay, there is some mistake in the spelling here. Thick and tough. Thick and tough is glycocalyx present outside the bacterial cell wall is called capsule. Okay, slime layer, capsid or capsule, or it is the papulos. It is the capsule. In some cases, in some bacteria, it's not so hard and tough. Okay, it is like some slimy. Okay, and it is mucus-like and it's called slime layer. Okay, but in some cases, it is dry and hard, rigid and tough, and that is the capsule. Okay. Now we come to the eukaryotic cells. So about the prokaryotic cells, it is already finished. Okay. Now in the next class tomorrow we will start with the eukaryotic cells. But before that, I will just want to recap and tell you some important points. That what we will study today is about the prokaryotic cells, and we know that prokaryotic cells, the bacteria, the mycoplasma, the blue-green algae or cyanobacteria are the examples of the prokaryotic cells. Okay, so in case of the bacteria, we have got 4 different types based on the shape. Okay, there is a bacillus, rod-shaped, the focus is spherical. Okay, and the comma-shaped vibrio and the spiralum. Okay, spiral-shaped spiralum. So these are the types of bacteria based on the shape. Okay, then we studied about the the okay. Now, there are the important point to be noted here is that apart from the genomic DNA, there is another DNA, circular DNA present in the cytoplasm of the bacteria, that is the plasmid, and this plasmid is very important with regard to biotechnological processes. Okay, biotechnological, this forms the biotechnological tools. Okay, that we studied in class 12. Okay, and now. Ribosomes are present in both eukaryotic and prokaryotic, but ribosomes is the site of photosynthesis, and mesosomes are there in the bacterial cells, and that is the extension of the plasma membrane into the cytoplasm, and that is responsible and that got some important functions. Okay, and it helps in respiration and also some kind of synthesis, secretion, etc. Cell and modification in prokaryotic, we find that there is the kind of tightly bound 3-layered structure, and these are the glycocalyx, cell wall, and the plasma membrane. So innermost is the plasma membrane, then the cell wall, then the glycocalyx. And again, there is a division of the based on the bacteria, based on the chemical composition. Okay, and envelope. Bacteria can be divided into groups. Okay, and based on the response to staining, they are gram positive and gram negative. Whether this bacteria will take up the gram stain, then it is gram positive, and whether the bacteria will take the stain or not. Okay, a gram positive bacterial covering is little simpler, because as we observe here, we don't find the double layer of the lipoprotein. Okay, the plasma membrane is there, then the plasmic space, and then the cell wall peptidoglycan, middle of the peptidoglycan. But in case of the gram-negative bacteria, there is a double layer. Okay, and in case of this, and they are not taking the the stain, and are called gram. Glycocalyx could be loose, like I told you, mucus like, okay, and can be tough, thick, and they call the capsule, and this is. They provide, cell wall is actually the structure it provides and defines the shape of the bacterial cell. Plasma membrane is semi-permeable, and it has some extension. Okay, and it has the mesosomes may be it is the extension of the plasma membrane, and that will be in the form of the vesicles or tubules or lamellae. Okay, so it is different forms of the mesosomes. The functions are, I told you already, in respirations and DNA replications and distributions, secretion process, and to increase the surface area, and it also contains some important enzymes for the metabolic reactions in the bacterial cell. Some are the pigment containing structures are present in the extension of the cell membrane, and it's called chromatophores. Okay, and now, in case of the movement, some bacteria, some are motile and some are non-motile, and those who are motile, they have got flagella. With the help of with the movement of that flagella, they are able to move, and this flagella has got different structure, different types, and depending on their composition. The composition is the filament, hook, and basal body. But whether they are present all over the body or they are two in number, so bacteria are grouped. Okay. But if you talk about the composition, the filament is there, hook which is there in the. In case of this is about the here you can see this is the gram negative. This is the gram about the gram negative bacteria, and this here it is the gram positive bacteria. Okay, so in both the bacteria you can see the hook is there. Okay, and then the basal body, and then the filament is there. Okay, now in case of some bacteria we find apart from the blood, there is the other extensions from the surface, that is the pili or fimri, which are not involved in the motility but involved in other functions. Okay, so that is the fimri is like it is a bristle like fibers. Okay, and it helps in the attachment to the rocks in the streams and host tissues. Okay, ribosomes are of size about 50 nanometer by 20 nanometer. Okay, the ribosomes are smaller than the ribosomes of the eukaryotic cells, and it has 50s and 30s units, which forms 70s altogether and present together. Okay, so several ribosomes they form an mRNA. They form like They are arranged in a chain fashion, okay, and this is called the polyribosomes or polysomes. And inclusion body is the structure which contains some important reserved food material. They have no any membrane, but they are lying freely in the cytoplasm. Gas vacuoles are found in blue green and purple green photosynthetic bacteria, and these are all about the structure of bacteria, prokaryotic cells. Okay, so prokaryotic cells means we have got the bacteria, blue green algae, cyanobacteria, mycoplasma, and pipiolo, pneumonia like organism. Okay, so this was all about the bacteria and its structure. Okay, tomorrow we will study about the eukaryotic cells and then the plasma membrane, different parts of the eukaryotic cells like plasma membrane and other cell organelle. Okay, so tomorrow we will again meet at the same time. Okay, after this class I will send you the DPP, and that you have to go through that DPP and you have to answer the questions and send back to us for our evaluations. Okay, so tomorrow again we'll meet at the same time, and thank you for joining today. So see you tomorrow at the same time. Okay, and till then, goodbye and take care.